All right, so in the last lecture, we basically talked about the sugar structures, especially focusing on the simple sugars and there are different types and also there are epimers and anomers and stuff like that. So we're going to move on to the bigger complex now of the sugars, basically, like above the disaccharides. So it's going to be oligosaccharides, polysaccharides, and so on. And also, we're going to talk about the glycoconjugates, basically the proteins that are conjugated or connected with the sugar, simple sugars. All right. So, like I told you, we're going to talk about the polysaccharides and some oligosaccharides today too. When you look at their structures, they can be categorized into two groups. One's let me just get to homopolysaccharide or homoglycan, where you basically have the same sugar, simple sugar as a monomer, and then you can either have the unbranched homopolysaccharides or the branched one too. And another one in another group is the heteropolysaccharides, also called heteroglycans. And the glycan is basically the same thing as the saccharides. So multiple sugar molecules connected to each other. And for hetero, as its name refers, you can have the more than just one simple sugar as a unit for the polymers. So you can have two of them like alternating, or even three or four or so based on the what kind of polysaccharides you have. And also you can have unbranched and branched one too. They are involved in a lot of biochemical reactions and the functions. Basically, they're the big like a storage entity where you can actually find the starch and glycogen as an example of those like energy storage molecules. And also they're involved in structuring the cells, like cellulose in the plants and the chitin in the crustacean, basically, are the bigger like sugar molecules, polysaccharides that are involved in such processes. And here are some actually examples of the cellulose, glycogen, as well as the starch that we actually discussed last time. And for the starch, you can actually find the amylose and amylopectin too, of the sub units of them. And the cellulose and the starches are found in the plants and basically serve as a storage of your sugars or the energy source. Although they have a slightly different subunits, like beta glucose or alpha glucose, but their bonding is pretty much the same as 1 to 4 connection. Of course, I mean, this will be beta 1 to 4, and for the starch, it will be alpha 1 to 4 connection. But amylopectin can actually branch, so it can also have 1 to 6 connection. Excuse me, I'm talking about the carbons within that, the 6-member ring of the sugar. We talked about it last time, right? So I'm just, not explaining too much about that. But anyway, you can have a branch using this 1 to 6 connection linkage. And the same thing applies to glycogen here too. Going back to the functions that the polysaccharides are having, it also involved in the bacterial cell wall, and an example is peptidoglycan that we're going to actually discuss its function and the structure too within this lecture. But this is a crucial part of the cell wall for the bacteria. And also the polysaccharides are involved in recognition during the cell signaling pathways. So many polysaccharides are located on the cell surface and kind of work as some kind of specific region or the recognition site for the cell-to-cell interaction or some kind of triggering of the cell signaling pathways. All right, and again, we're going to talk about these shapes and other complex, like structures, in short, basically. So the first function of the polysaccharides that I said was the storage. And why? Because you can basically use just the simple sugars as a storage too, right? And it's readily accessible because you already have a single sugars. So you can always break it down to make it into a different molecules, or basically get the energy from using the internal energy that is saved from that sugar molecules. But the living systems do not actually keep them as just a single molecule, single simple sugars. And the reason is because it raises the osmotic pressure tremendously. Because imagine that we talked about the colligative property of the water, where you have some amount of molecules. It'll actually change a lot in terms of like boiling, or the melting temperature and so on, as well as osmosis. And that is really important, especially for the cells, because they basically have the plasma membrane where it keep the cytoplasm, the internal stuff in the cell. And when you have a differential osmotic pressure build up inside and outside of the cell, then cell can basically shrink down or burst out, right? So it has to actually make a nice balance for that osmotic pressure. And if you're adding too much like sugars in there, lots of like little molecules, basically you're raising up the osmotic pressure a lot, will cause the water, all the water flows into the cell, and then will cell's gonna just burst out. So to avoid that scenario, you can actually use the polysaccharide, where it still can hold lots and lots of the simple sugars, but it's in the connected form. So instead of having like tens of thousands of little sugar molecules, now you're gonna have only one chunk, big, huge molecule, which is polysaccharides. And it won't skew too much of the osmotic pressure, because osmotic pressure, or all the colligative properties, are about the actual total number of solute molecules in the solution, not the type of it, or the weight of it, or the size of it. Does that make sense? So you can have as many as 10,000 simple sugars within just one polysaccharides. So that's why it is a really convenient way to store all these little sugars. And again, starch is an example here, where you can see like a long chain as well as some like branches here too. All right, so now we're gonna go over all different types of polysaccharides one by one, basically, and we'll discuss their structure, their usages, their functions in the cell, cellular like living systems, and the type of linkages and so on. So it's all kind of biochemical, facts basically, but are really important. The starch is found in plants basically as a storage form of the energy, and its most common storage form, or the polysaccharides in plants. So you can see them in pretty much all different kinds of plants, like potato, corn, wheats, and like everything basically, whenever they try to live and save the energy, or the sugar. And when you look at the starch, actually, it is a mixture of glucose polymers, and there are 20 to 25% of alpha amylose. And here's a structure of the alpha amylose, where you have multiple glucose, and it has the alpha 1 to 4 linkage. And as you know, alpha, tells us the anomer. So whether this OH, the hydroxyl, is like facing like pointing down or up of the this sugar plane, right? So since this OH, supposedly OH, is in the pointing the opposite direction from this exocyclic carbon, you can call it as alpha. We talked about this last time. And there's a C1 to C4 linkage. So it's going to be alpha amylose, but basically 1 to 4 linkage. And that makes the really nice long chain. And we'll also have the left-handed helix. This is an example of that. And as you know, left-handed helix is basically, use your left hand and just wrap around the helix. And if that kind of wraps nicely around, and your, all the fingers are pointing, especially index finger pointing upward, then it is the left-handed helix. And also the rest of it, like pretty much the 75% to 80% are the amylopectin. And when you look at the amylopectin, it basically has the same linkage as the alpha amylose, where it has alpha 1 to 4 linkage. But it also have alpha 1 to 6 linkage. And C6 is, again, exocyclic carbon. So instead of, it makes the linkage with the sugar ring, it will actually make a linkage to the one functional group that sticks out of the ring. So that it allows the making that kind of branch kind of structure there. So, and this will happen every 24 to 30 residues. And think about it, you can have like tens of thousands of simple sugars connected to each other in starch, right? So you're gonna have a lot of branches. And for the, each branches, you can have more branches, basically having a really kind of complicated structure, like a web-like a structure there, can store lots of, glucose. All right? So this is an example of the alpha 1 to 6 linkage. And as you can see, here's the exocyclic, carbon, and here used to be the hydroxyl that is now deprotonated and get and also hydrated basically to get this linkage here. All right, so I mean this is really convenient, like I told you, in a way that we can store lots of energy without changing too much of osmotic pressure there, which is great. But also it's a little bit complicated structure, meaning that there are a lot of enzymes or the proteins have to be involved for the digestion of this, complex polysaccharide. So it's basically a team effort, meaning that there are several steps with the many different kinds of enzymes that are involved for breaking down this complex structure of the polysaccharide. There are some examples here. The first one is amylase. Amylase in saliva that breaks the some alpha 1 to 4 bonds within the starch. And then, so you're gonna have a smaller chunk, basically, sort of like oligosaccharides. And then another one in the line is amylase, and that is excreted from the pancreas and gets into the small intestine actually. And then it'll work to break down the alpha 1 to 4 linkages again, but more finely to get the mostly into disaccharide form, which is maltose. So you can see it here. Here's a alpha amylose that you have like that nice chain kind of structure here. Here's the alpha 1 to 4 linkages. And then this pancreatic amylose break everything down into these two disaccharide units where two glucose are still linked together, which is called maltose. And another important enzyme is the alpha glucosidase. Basically sequentially remove the one residues at a time at the alpha 1 to 4 linkages. So it's going to make it into the single glucose. Now you can actually use them as some kind of converting into the other molecules or just getting the energy out of that. And also there are debranching enzymes that are working to cut this alpha 1 to 6 linkages, and that is the bond between the main sort of chain alpha amylose as well as the branches. Okay. Any questions so far? It's just a lot of information, right? Nothing really complicated. The next polysaccharide that we're going to look at is glycogen. And that's mostly found in, not all the time actually, found in animals instead of plants, but serves the same purpose as starch, where you store the urea energy. And it is quite a lot in the animal too. So it's prevalent in its weight, like 10% of liver is basically consists of the glycogen, and your muscle too, you can find 1 to 2% of total weight is just the glycogen there. And you can imagine that, right? Because it requires a lot of energy, liver too, lots of energy, or you also convert it into the storage form. So that's where you can see the glycogen a lot. And it's when you look at its structure, it's similar to amylopectin, and amylopectin is again has a long chain of the glucose, and then you have lots of branches. But the frequency that you can see that each branching points are about 24 to 30 glucose units, right? As opposed to this glycogen have the more finer basically branching points there. So you can see every 8 to 12 residues, and residues I'm talking about the single sugar, the glucose molecule, right? You can see that that is branching happening. Again, using the same linkage, alpha 1 to 6 linkage. Question? Yeah, so that is more like random, basically, like stochasticity. Just that sometimes happens, sometimes not. It depends on the enzymes that are involved there too. So, and in animal, the enzymes that are sort of prevailing for making this glycogen is different from the enzymes that are involved in making starch. So that's the difference. But you're not going to see the like really kind of regular patterns there at all. It's kind of random here and there. Same thing applies to the branches of the branch. So you're going to see that randomly. And it depends on how many glucose molecules you have too. So, all right. And this glycogen can be quickly metabolized when blood sugar gets low, because it has lots of branches, so there are a lot of ends, right? Basically, branching ends, and the end of each chains that you can basically attack and detach the single sugars there. So same thing applies here too. So you have a highly branched structure there. That means you have lots of ends. And interestingly, all the branching ends are non-reducing ends. So we talked about reducing ends and non-reducing ends for the polysaccharides, right? And the only difference there is that whether it can make another linkage with the other sugar molecule or not. So if it is possible, then it's reducing end. If it's not, that's non-reducing end, right? So and that means that although you have lots of branches in glycogen, it's not going to be able to extend it because those are the non-reducing ends, as opposed to you're going to have only one reducing end in the entire glycogen molecule. And that's the end of this main chain. So it can still grow, and it can still build the branches, but all the branches will not grow, essentially, unless you have other enzymes are involved there to make it into the reducing end form. Anyway, this is like almost like too much detail, but anyway, you're going to have a lot of branching ends, and that means you can easily mobilize, and quickly mobilize the glycogen. And glycogen being mobilized is basically you break it down and make it into the useful form, where it becomes the disaccharide or the monosaccharide. There are two classes of enzymes that are involved for mobilizing or metabolizing the glycogen. One is glycogen phosphorylase that cleaves the alpha 1 to 4 linkages from the non-reducing end. So it'll attack all these branch points, basically, end of the branches. And another class is the glycogen debranching enzymes that will attack the alpha 1 to 6 linkages. So it'll cut, chop down all the branches there, basically. All right. And glycogen is not a fat. Question. Mm-hmm. Why would they do that? Why? Why? Oh, wow. I remember you that you also asked some kind of philosophical question last time. So I think it's more like a philosophical question. No, no, this is a great question. I think why would you want to break down glycogen? Yes. So I mean, it's precisely. So glycogen is really nice for storage because it won't skew the osmotic pressure at all. But you can't use it. It's just the one blob of sugars. You have to just break it down to actually use it as an energy source. And we're going to talk about the metabolism later on. But basically, you have to start with the single sugar molecule, and then it'll be broken down to basically water and carbon dioxide, and then you're going to get a lot of energies from that. So, but you can't do that with the glycogen, which is a polysaccharide. So that's why. All right. Yes. Yeah, it can be confusing, right? Um, yes. So I actually explained a little bit, and I will do that, but I won't dwell on it because I'm not going to ask about it, in the exam. But anyway, um, reducing end. So instantly, you guys, lost your interest now, right? Because I just said that, it's not going to be on the exam. But the reducing end, I wish I could go back to the previous one. Reducing end is basically the end of a polysaccharide where it ends with the hydroxyl. And you remember that there's an anomeric carbon in C1, right? And that is important for making linkage with the other sugar molecule. If it is just has its intact form as having the hydrogen atom and the hydroxyl, then it can always lose the proton or the hydrogen and can make the glycosidic linkage with the other sugar molecule. So it's reducing end because it can be reduced and can make other connections. As opposed to the non-reducing end is the think about sort of in the middle of the polysaccharide where you have only oxygen molecule that is holding to the two simple sugar molecules. Because hydroxyl that was attached to the anomeric carbon is now all reduced and you lose the proton and then already made the connection to the other one. So there's no way to make new connection with the sugar at all unless you lose that sugar and get it recover back the hydroxyl group, right? So that's a non-reducing end. So those are the differences. Okay. And the again, glycogen is a polysaccharide. It's not a fat. Just a second. So don't be confused. I'm just saying here that the most of the energy in the living system is stored as a form of fat, and glycogen is not a fat. It's just the polysaccharide. So it serves as a more like a temporary storage method, as opposed to fat is pretty constant, like pretty stable. And it is most efficient form to save energies because it takes up the less energy, less weight and less space per energy, which is in kilocal. But it also has a downside where it takes a much slower time to metabolize, right? So if you do the sort of aerobic exercise, you have to do that for at least an hour or so to start burning your fat. Otherwise, you're just burning your glycogen. That's all, which is a temporary form. So you're not going to lose your weight. If your purpose is losing weight for your exercise, same thing applies to starvation, right? You just skipped your lunch or dinner for losing your weight, but you have to wait until like 18 hours or so at least to start burning your fat. So it's much less efficient. But again, really efficient in a way to store the fat. Question? how they are related to each other. So the question was that we learned carbohydrates, right? Which are basically saccharides, polysaccharides, monosaccharides, and things like that. How they're related to lipids. When you look at the structure, we're going to talk about lipids actually, I think, next lecture, next two lectures. Their structures are vastly different. But there are ways to convert one to the other, at least indirectly. So they are not directly related, to be honest. They go through the different metabolic pathways and so on. They're used in the different regions of the cells, have a different functions and so on. So they're seemingly like loosely related, but those are really important because those are the two major energy storage forms. So it's like you take up the energy from the fat and make it into the saccharides basically, and take the energy from the vice versa to from the saccharides and to the fat for the long-term storage and so on. So other than that, nothing much. Question? Were there any questions? I think I've seen raised hand, but okay, I'll just keep going. Oh yeah. You forgot. You forgot what? Oh, catabolism. What is catabolism? Can anyone answer to this question? What is catabolism? Yes. Is what? Exactly. So, I mean, it's basically the chemical reactions, biochemical reactions. So it happens inside the cell to break down the molecules to get the energy. Is that what you're saying? And there's anabolism too, which goes backward, right? And if metabolism is basically both of that, catabolism and anabolism. So again, we're going to talk about that much later, when we are talking about the metabolism. But all right. So cellulose is another type of polysaccharides that is used in plants. And this is also the most abundant natural polymer in nature. So it takes up like up to 80% of dry weight of a plant. So when you actually eat plants, veggies, you're eating lots of cellulose, basically. And the cellulose is the major component of a plant cell walls. And plant cell walls are basically for fighting against the osmotic pressure that's built up, as you know, because we talked about it. And this cellulose has the property of rigidity. So it confers the rigidity and mechanical strength of the cell wall. So that's why plant is some kind of quite rigid, and can resistant and resilient to some environmental stresses like osmotic pressure. And its role is about the structure, stabilizing the plant structure. So we talked about this already, and it has the homopolysaccharide, and has the linear structure where its subunit is beta D glucose, and it has the beta 1 to 4 linkages. So you can see the linkage here, looking similar to the glucose or the starch, right? Basically, but the glycogen, but the only difference here is that it has a beta linkage instead of alpha linkage, but it still uses this glucose, and this can get as long as 15,000 units. And once you have this kind of long chain, which is a linear structure, they can be staggered, like stacked, but in a staggered manner. So just like here I said, bricks in the wall. So instead of having the really kind of matched kind of linkages, they're going to have a staggered manner, will strengthen even further of that structure. And then they can form this kind of sheet. And these sheets can also link together, basically, to even fortify this structure to make them really rigid cell wall. And for these interactions, there are the hydrogen bonding having a critical role there. And there are three different types of hydrogen bondings are involved for this sheet-like structure of the cellulose. Intra-strands, because you have lots of hydroxyls and hydrogen atoms. So you can basically make them, here's a hydrogen atom, and that you can't see, and here's a hydroxyl, right? Between them, there can be on hydrogen bonding there. So this is called intra-strand H bonding. And also you can have it between these strands or the sheets. So interstrand, intersheet, hydrogen bonding. So that all makes the really this kind of strong structure. And it'll be resilient to hydrolysis, basically, and is insoluble because they're so well packed together. There's not much of room that water molecules can get in, or the other enzymes, the proteins can get in and do something there. It's just a really well packed and organized structure. Yes. That is a good question. So the question was that what about the water molecules? Can they actually those water molecules compete to these hydroxyls for making hydrogen bonding? If that is possible, then it should be a little more soluble, right? But that's almost impossible because this cellulose fibrils or the sheet-like structure is so packed together. And again, it has the staggered form. So it minimizes any kinds of space between the sheets, strands, and things like that. So there's no room, basically. And that's why plants can absorb, water, but only on the specialized regions like roots and, right? Shoots and things like that, because rest of them, even if cellulose have lots of hydroxyls, they're not accessible, simply not accessible. Another one is chitin that has the similar structure to cellulose and function too, basically. It's about the structures, making the really kind of strong structures and for their integrity. And you can see that a lot in the exoskeleton of crustaceans, insects, and spiders, and so on, as you know. And it has the extended linear chain, the linear structure, again, like the cellulose or the starch. But instead of having glucose, it has the N-acetylglucosamine or GlucNAc. So gluc was from this glucosamine, and NAc was from N-acetyl. It's kind of weird name, but it's widely used. So GlucNAc. And when you look at the structure, the molecular structure of the GlucNAc, and make a comparison to glucose, the only difference is this moiety here, NHCOCH3, which is attached to the C2 instead of hydroxyl. And it has the beta 1 to 4 linkages, so it's going to be really a long, again, linear structure without any branches there. But it will have extensive hydrogen bonding, similar to what we've seen with the cellulose. And here's an example where it has the intramolecular hydrogen bonding, where here's an O and here's H. They're having the H bonding. And we're kept, basically discussing all different kinds of polysaccharides here. This is, again, a quite important polysaccharide, which is called glycosaminoglycans, or GAGs, and also known as mucopolysaccharides. So those are basically all the same thing. But it's long, unbranched polysaccharides consisting of a repeating disaccharide units. So far, for the chitin, starch, cellulose, glycogen, and things like that, we're talking about the single unit as a simple sugar, right? Glucose, or the GlucNAc and stuff like that, as opposed to now we're talking about the polysaccharides that have a unit of disaccharide, two sugars, attached together. So it's gonna be a heteropolysaccharide, basically. And when you look at its structure, it has hexac or the N-acetylhexosamine, which you can see the structure here. And the another one within this disaccharide unit will be either hexose or hexaronic, hexaronic, hexuronic, hexuronic acid. Sorry. And this hexuronic acid can also be sulfated, meaning that one of its functional group can contain sulfur atoms. And we already know that this sulfur atom does a lot of interesting things, including making bonds in the cystic cysteines, two cysteines, and so on. So we're going to talk about that in a minute. Yes. That is correct. So basically, the question was that is this repeating disaccharide unit is the same as the heteropolysaccharide that I mentioned. It's the same thing. The GAC is one of those heteropolysaccharides, but heteropolysaccharides can have three, like a trisaccharide as a unit too, or tetrasaccharides as a unit too, as opposed to this GAC, the glycosaminoglycan, always have disaccharide as a unit. So that's the difference. So again, one's hexac and the others can be either hexose or hexuronic acid here, and that can be sulfated too. And since you can have, there are different types of hexac and different types of hexose, of course, hexose. It can be anything with the 6 carbons, right? And the hexuronic acid too. You can have like tens of different kinds of, GACs basically. So it's actually says here. So depending on the members of these guys, you can have many, many different kinds of molecules, and also depending on what kinds of glycosidic linkages you have, 1 to 4, 1 to 6, 1 to 3 sometimes. Here you can see it 1 to 3, 1 to 4, sometimes 1 to 6, and also it can be alpha or beta linkages too. Different anomeric, anomers basically are used as a single unit. So you can have lots and lots of different kinds of glycosaminoglycans. And it has a very high density of negative charges. And you can imagine that, right? Because it has containing the carbonyl group, as all the sugar has, like CHO or the ketone group, where it can have the negative charge there, as well as it'll have a carboxylic acid or carboxylate group from the hexuronic acid. So it's acid, so you're going to have the minus, of course, once you lose the proton, right? So lots of negative charges. And also, you have a sulfate group here too, which is like imagine the sulfhydryl group SH, and then you lose this H basically, proton, then you're going to have negative charge there too. So you're going to end up having lots and lots of negative charges built up within this GAC molecule. All right. And these are different types of GACs basically that we're showing. And these are the different type of disaccharide units that you can use for building the glycosaminoglycans. So you can use hyaluronic acid, which you've seen actually in the previous slide, or the keratin sulfate or the heparin, or many other things basically. And that basically will confirm different properties of GAC molecules. Here's one example where GAC is used. I always have trouble with putting this at the right angle. So, one example is that the GAC is used in the ligaments or the cartilages on the connective tissues in conjunction with the collagen fibers. So in the extracellular spaces of connective tissues, and connective tissues are cartilages, tendons, ligaments, and whatever that are connecting to different kinds of tissues, bones, and muscles and stuff like that, as well as synovial fluid, which is a sort of fluid that is lubricating the joints and the vitreous humor of the eye. You can imagine that all of them are pretty like a viscous, right? Kind of making the kind of like viscous liquid that is can make the long kind of thread or the thread like a structure there. Um, uh, it's all because of these GAC molecules. It consists also it consists ground substance in ECM, and ground substance is just a term for the certain tissue structure there in the ECM that connects the collagen fibers. So hopefully you remember the structure of the collagen, where it has the helical structure from the polypeptide, and then basically three helices will get together to form this collagen like fiber units, and then these guys will get together and make the actual collagen fiber. And to make this structure even stronger and also stabilize in this glycosaminoglycan molecules are surrounding this collagen fibers gets into that too. The GAGs found in the collagen fibers have 250 to 25,000 residues long, so it can be highly variable but pretty big, right? And again, it makes the slimy mucus-like a structure, so it will confer this property of a viscosity and elasticity, and that is important for the collagen fiber, as you can imagine, as well as all the connective tissues. You have to have a flexibility there, right? So that's how you can get that property. And since GAGs are all highly negatively charged, it can easily bind the cations like potassium, calcium, sodium, and so on. And it can be highly hydrated too, so it can take up to the 1,000 fold of the volumes compared to the non-hydrated form, because it has makes the really complicated structure there that we're gonna see, I believe, in the next slide. And this degree of viscosity basically depends on the degree of shear stress because of all connections on stabilizing these collagen fibers, as well as other structured higher order structures by the GAG molecules, and works as an excellent lubricant and absorbers. Yes. No, it's okay. Yes, that is true. So, and that could be important, right? So instead of having the same kind of viscosity, if you have the higher shock, then you want to have the higher like viscosity, or the sort of behaving in the slightly different way that you can protect your tissue more efficiently, right? So because it has the really complicated structure, it'll behave slightly different depending on the how you're going to give the force on it. So, all right. So now we're going to move on to the glycoconjugates, which is a protein, and the with the sugar decoration basically. Any questions so far in terms of polysaccharides? All right. So the glycoproteins are basically proteins with the sugars coated on the surface of the protein, and that is really important for the targeting of the certain like within the cell signaling pathway, or sorting those proteins, recognition of a certain site, certain type of cell, and the protecting from the degradation of that protein that is decorated with the sugar too. And these modifications are not encoded by or in the DNA, because DNA only tells about the amino acid sequence, right? So it's not going to tell anything about the decoration basically on those proteins. So it can be highly variable depending on what kind of environments in those proteins or cells are under, or also what kind of stress it gets, what kind of developmental stages they are in, and so on. And that gives the high degree of heterogeneity of these glycoproteins. And that is a great thing, because now you have a flexibility that can respond more quickly to the depending on needs. And there are two types of linkages between the sugar and the proteins for building the glycoprotein: O-linked and N-linked glycoproteins. All linked, as you can see here, have the oxygen for the linkage between these two different proteins and the sugar. And that happens on the hydroxyl group of serine and threonine. And as you can imagine, if you lose the proton here from the hydroxyl of the serine or threonine side chain, then now it is available to make a linkage to a sugar molecule. And for N-linked ones, asparagine is the one that can form these linkages because it has this amino group in the end of the side chain, so it can be used for making the glycoprotein. And here are some examples for the O-linked saccharides or O-linked glycoprotein. It can use disaccharide or monosaccharide or basically pentose or the hexose. It really kind of depends what kind of things it can interact with. There are a lot of sugars can be decorated basically on the protein to form the glycoproteins. Same thing applies to the N-linked saccharides or the N-linked glycoproteins. So it can actually have a really kind of elaborate structure like this, where you have different kinds of sugars are involved there, different kinds of linkages, as well as branching too, right? So it's quite complicated structure. These are the examples of the sugar decoration basically on the glycoproteins. So you can have GLUC-NAC, mannose, different numbers of them, to form the certain protein. It's ribonuclease B. Human immunoglobulin G has the branchings too here, mannose, and then GLUC-NAC and stuff like that. And these are all the different kinds of lysosomal enzymes and different examples. Well, I'm not going to ask, how many mannose, how many GLUC-NAC you're going to have in the ribonuclease B. But at least you should understand these are there are different types of sugar decoration. I'm just keep saying decoration, but it's actually glycoproteins, right? O-linked or N-linked proteins, and what type of amino acids are involved there for making which kinds of linkages are important. So here you can see asparagine for all of them. So you can imagine what kind of linkages are involved there. Here's another example of the glycoproteins, where different types of sugar molecules are attached to the amino acid that are becoming the glycoprotein. And this is on sort of a pattern of the sugar decoration in the erythrocytes. And as you probably know, erythrocyte is basically just a red blood cell. So, and you can have like A type, B type. Is it type A, type B? Like AB or O type, blood. And that is because you have different kinds of antigens or on the surface of your red blood cell or erythrocytes. And it basically first has the H antigen as the precursor, which has just the GAL and the connection through the beta connection 1 to 4 linkages and GLUC-NAC, and then you're going to have your protein, glycoprotein there that is presented or embedded on your plasma membrane of the erythrocytes. So it can be present like that first, and then there are going to be additional sugar attachments, and based on what kind of sugar that are attached, you can have type A or type B of your blood, or O or AB basically, where you have GAL or galactose, or GalNAc for the N-acetylgalactosamine that are attached there. It's a small difference, as you can see. It's not even the different sugar. It's the same galactose, but whether you have hydroxyl or the amino group there, right? Even if it's a small changes, a small little decoration, you can still make a huge change there, where you can't get the blood from the A type if you're B type, and so on. So you can just imagine that it's a simple decoration of the sugars, but it's really, really important for recognition and for some biological reaction biochemical reactions. Okay, so and we're moving on to the different type of GAGs now. Here's a proteoglycans, and name kind of doesn't mean too much, right? Proteo is protein, and the glycans are saccharides, and so it's pretty much like a glycoproteins, but it's actually a subgroup of glycoproteins, and it has to contain glycosaminoglycans or GAGs. So whenever you see the glycoproteins that are containing GAGs, then you can call them proteoglycans. And again, like we discussed, in the section of GAGs, there are mainly found in the cartilages, in the joints, where it has the collagen fibers and the proteoglycans or the GAGs there. Again, these are the different examples, and so you can basically give the different properties or the different types of GAGs there. And this is what you actually can see as a GAG shape. So it looks like some kind of beads linked together, but it actually has the one core thread or the core chain there, and have a lot of branches, and each branches have some blobs there. So this is a cartoon actually shows this structure here. So proteoglycans basically has this backbone, which consists of a hyaluronic acid polymer. So it's always a hyaluronic acid, and you can actually see it's not here, but the structure when you go back to the GAG, the slide that we discussed GAG, you can see the actual molecular structure of the hyaluronic acid there, which has the both carbonyl group and the carboxylate group. And these like a bristle-like structures are attached to this backbone, and they are basically core proteins right in the middle, and there are lots of like GAGs that are attached to this core protein. So overall, it looks like the kind of bottle brush-like shape there. And it is very highly hydrated. And you can imagine, right? There are a lot of like little branches there, and all the water molecules can be hidden basically or absorbed in those like little like polysaccharides or the oligosaccharides. So you can actually it can contain lots of water there, and it has a high resilience too because of this property of containing lots of water molecules. So simply if you press it down and squeeze it, then you can squeeze out the water, and if you release it, then you're going to gain the water back in this GAG or the proteoglycans, and that's again gives the flexibility of your tissue, your structure. So it's really useful property feature for the joints, and it's also, this property is responsible for the nourishment of a cartilage in joints where there's no blood vessels. So you have to have another way to convey the water and the other energy-based sources there. And this property basically can be utilized for that process. Any questions so far? Okay. And another type of glycoprotein is the peptidoglycan that forms the bacterial cell walls. And I don't know if you know this or not, but there are two different, mainly two different types of bacteria, gram-positive and gram-negative, based on the whether they have outer membrane or not. And regardless of gram-positive or gram-negative, you're going to always have cell walls. And this cell wall consists of these peptidoglycans, which is really critical for the protection in the hypertonic medium. Again, it's all about the osmotic pressure, right? So you have to just keep your stuff, like cytoplasmic stuff, inside the cell, but you don't want to burst out your cell. So to control that, because you generally have more stuff inside cell, you're going to have osmotic pressure build up inside to diffuse in the water molecules into the cell. So eventually you're going to have a burst out your cell. To avoid that, you need to have a really strong kind of like case or the block that's surrounding the cell membrane, which is a cell wall here. So it applies to the plants as well as bacteria too. And also it is responsible for virulence of bacteria because it contains lots of other proteins, glycoproteins, and so on. Um, Basically serves as gives its own property as well as binding regions for with the host cells. It has the beta 1 to 4 linkages, and it uses a disaccharide as a single unit, and it has a Gluck neck that we talked about. Instead of having the hydroxyl, it has this amino like group. And also there's a NAMA, which is N-acetylmuriatic acid, and this mura or the muras means its wall. So it's basically named because of this has been used for the cell wall here. And also have these kind of amino acids that are attached to the end of this NAMA, which are alanine, glutamine, lysine, and alanine. And that is important for making the like cross links of these peptidoglycan polymers. So how it works is that this disaccharide unit makes the really nice long chain, but just a long chain doesn't do anything too much. I mean, we can imagine basically the cellulose structures where it has all the cross-linking, using the hydrogen bonding and so on. So same thing applies to this what is it? The peptidoglycans in the bacteria too, where it uses those tetrapeptides that I talked about, alanine, glutamine, lysine, and alanine, which is depicted here as this kind of beads here in the blue, or four of them here you can see, and this one can be connected to the other tetrapeptides that is attached to the other chain of this peptidoglycan. And these two are connected by this pentaglycine bridge, which is simply just literally GGGGG. So that connects these two each other. And this connection is really important. Again, makes the cross-link of the different chains of peptidoglycans to make this bacterial cell wall really rigid, right? But one of the most important scientific discovery in human history is finding the penicillin. And penicillin especially destabilizes this cell wall structure. And how it works is that this penicillin, which you can see the molecular structure here, gets inside the active site of this enzyme called DD transpeptidase. And this DD transpeptidase make the linkages between the different chains of peptidoglycans by making those like GGGGG link, the penaglycine linkage there. So if penicillin gets into the active site of this enzyme DD transpeptidase, it won't be able to bind to its original substrate here, the tetrapeptide that are linked to this peptidoglycan. So you won't be able to make these linkages. Now this cell wall, bacterial cell wall, will be kind of loosened up and will degrade it over time. So it won't be able to survive. All right, so this is the last slide actually. And these are the list of things that you need to memorize. That doesn't mean that you have to memorize all of these things precisely, but at least you need to understand the structure. You need to be able to tell that this is what it is. You don't have to draw that, but at least have to identify those, like the linear polysaccharide structures, glucose, galactose, mannose, fructoses. We already talked about this because these are important for telling the epimers, endomers, and so on, right? So some of them are really critical. And the cyclic ones for these, are basically the similar molecules too. And for polysaccharides, you should be able to tell that, what are the differences between different polysaccharides like cellulose, chitins, glycogens, starches. And we talked about the glycoproteins too, right? I gave you some examples of them too. Should we understand how they are made, what kind of linkages there, what kind of amino acids are involved there. All right? And that's it. You guys are dismissed. Oh, questions? Sorry. Oh, just keep it on then.
Slide, I guess. So these are like disaccharides that are in the